Kelly. And what a wonderful presentation to start off the day. And I'll be picking up on some of the themes of that presentation. In my presentation, I'm going to be talking to you about the concept of durable resistance. Now, durable resistance is a very simple concept and a very powerful one. We've had advances in genetic analysis over the last 40 years since this concept was introduced by Roy Johnson that have allowed us to dissect out the genetic components of durable resistance in a number of sources that have been identified as being durable. And that's allowed us to improve our understanding of durable resistance at the level of the gene. But I believe we've still got a lot to learn when it comes to actually applying the concept of durable resistance in the landscape of agriculture. And what I mean by that, and this is what I mean in the title of my talk, at the continental level and even beyond. Now, to add to that, as I said, durable resistance, a very simple concept, but it is misunderstood at times, and it is actually applied inappropriately. It's a retrospective term. So in the strict definition of the term, in the strict sense, you can't breed for durable resistance. If you want to be very pedantic and follow the definition of durable resistance, it's impossible to breed for durable resistance. We only know that resistance is durable when the products of a breeding program are put out into agriculture and they stand the test of time. Now we have a number of examples of large-scale disease control, but I think, and certainly durable resistance has contributed to that, but I really don't think we fully understand why those large-scale, long-term disease controls examples that we have actually worked. There's still a lot to learn. So in my presentation today, I'm going to discuss durable resistance in the context of that landscape of agriculture, if you like. But before I do that, I'll just recap on this very simple concept. So there's the definition that Roy gave to durable resistance. We all know this, very easy to remember. I read it in all of my students' dissertations, and we hear it all the time. But it's important to remember that this is a descriptive term, all right? And if you read Roy's review in 1984, this is what he says. He really emphasises it. It is a descriptive term. So when we talk about prolonged, how long? When we talk about widespread, how widespread? When we talk about the environment being favourable, how favourable? All right, so all of these really are descriptive terms. Now, as an example of that, I can tell you that we know for a fact that the Australian environment is very, very favourable for the development of striped rust in wheat. We know that. But I can also tell you that any striped rust gene that would have been deployed between 1800 and 1978 would have been considered to be durable. Sounds pretty strange. Well, the reason is because we did not have the disease. Now, that is a completely ridiculous example, I know, but it shows, I think, that this is the descriptive nature of the term, and that there is a need for some common sense when we apply it, and some critical thinking in the way that it's actually used. It's also a term that includes two conceptual elements. And again, this was very clearly stated by Roy. The first one of those is that there may be any one of several underlying causes for durable resistance. There's no single model that explains durable resistance. So I think with the work that we've done to date, we probably do have a single model to describe non-durable resistance, but the work has also told us that there's no single model that describes the genetic basis of durable resistance. We've found examples of simply inherited, and also polygenic durable resistance that involves major and minor genes, or major or minor genes, or even both. In the case of the gene cloning work that Professor McIntosh referred to yesterday that's been going on, we're getting greater and greater insight into the actual genomic structure. And a lot of the genes that are involved in durable resistance that are being cloned, we think could be involved in downstream processes rather than actual pathogen recognition. But there is one example of one of these pathogen recognition genes, an NBS LRR gene, that is actually implicated in durable disease resistance, and this is a gene in rice, PI35 to rice blast. So there's an example of a gene that perhaps superficially at least we might think would be non-durable, but in fact in agriculture has proven to be durable. Okay, the other conceptual element contained within the definition of durable resistance is that it is resistance that has remained effective for a long time. And I think this is really important, remained effective for a long time, but may not necessarily continue to do so in the future. All right, so just because it's worked in the past, it's no guarantee it's going to continue to work forever. So we can go through some examples here. A very nice one that our previous speaker told us all about, this SR2. It's been described as the most important disease resistance gene deployed in agriculture. Is it durable? Well, we've not detected virulence for SR2. There have been times when genotypes carrying SR2 have been loaded up with inoculum, they have developed significant amounts of disease. Bob referred to the bushfire analogy yesterday. They've been singed, but we have no evidence of virulence for SR2. So yes, by definition, this is clearly a durable resistance gene. PG1, stem rust gene in barley, originally found in chevron barley, deployed extensively in North America for many, many years. But virulence was detected by our colleagues in the US way back in 1988. So this is a gene that is considered to be durable by definition, but for which virulence was detected. Another one is SR31. The gene, the famous gene, deployed extensively on the 1BL1RS translocation that was overcome by UG99. Again, by definition, we would say this is a durable gene, but virulence was eventually detected. I just want to tell you a little bit about another gene, SR26, and ask the question, is it durable? We've not detected virulence for SR26. It was introduced into wheat from Thinopyrum by Doug Knott in Canada, and it's only been utilised in Australia, but we've used it a lot. It was first deployed in a variety called Eagle back in the early 1970s, and we've put it into 30 varieties since then. And it's remained effective. And you can see from that graph, that's just the percentage of the acreage occupied by variety, or only 3 varieties I should say, Eagle, Kite and Harrier, carrying SR26 in Queensland, New South Wales where we are now, and South Australia, I'm sorry. So you can see that those varieties occupied occupied significant areas over a significant period of time. So by definition, we would say that SR26 has all the hallmarks of being a durable resistance. But if we look a little deeper into that issue, the fact is that since the mid-1970s at least, I mean certainly in the early 1970s there were appreciable amounts of stem rust inoculum. That was the last major stem rust epidemic we had in 72, 73. But since the mid-1970s, stem rust inoculum levels have been quite low in Eastern Australia. So we could ask the question, really, is that those low stem rust inoculum levels, have they been due to the use of genes like SR26? Is it really, truly, intrinsically durable? Or has it remained effective, SR26, simply because inoculum levels have been low? Or is it a combination of those two factors? I think I'd be very, very nervous about deploying a gene like SR26 in an area such as East Africa at the moment where we have lots and lots of stem rust. It's a gene that's been durable for us, but I think if it was deployed in an area with high inoculum pressure, it may not survive the test of time. Okay, so just a couple of, I suppose, summary points about durable resistance. There's clearly a subjective element in the decision to describe resistance as being durable or not. While we seem to have, we've done a lot of genetics and we're continuing to delve in at the gene level, and this information is telling us that we probably do have a fairly simple or a single model to explain the genetic basis of non-durable resistance, but it's equally driving home the point I think that Roy originally made, that there is no single genetic model for durable resistance. Durable resistance may not remain effective forever. So just because we have something that has been effective and served us very, very well in the past, there's no guarantee it's going to continue to do so in the future. And then finally, and this is quite important I think, that when we start talking about large-scale agriculture and the control of rust over very large areas, the factors that determine durability of resistance are not always simple, and I think there's still a big gap in our knowledge, in our understanding of why we can actually achieve large-scale resistance, and as I said at the outset, the contribution of durable resistance to that. All right, so if we start now thinking, that's durable resistance. If we now start to think about the larger scale picture, and now I'm starting to talk about regional and even continental disease control, we do actually have some examples where we've had large scale, continental level, durable, or I should say sustained control of plant diseases. Two very good ones of these, two very good examples of this, are powdery mildew control in Europe. So this was a problem, mildew in barley and in wheat, were two diseases that were big problems for many, many years, and there were a series of boom and bust cycles where single genes were deployed and overcome. In the last 30 years or so, mildew in wheat and in barley in Europe has been largely controlled. So we would say here's an example of large-scale, long-term disease control. One of them is due to monogenic resistance. This is in barley, the gene MLO, a single gene. The other, in wheat, the belief is that this is polygenic control, but two very, very different genetic models giving long-term sustained control of two very damaging pathogens. Another two examples, I think very good examples, and the first one of these, I guess, has its genesis way back in the work of McFadden that was covered very nicely in the previous presentation. Stem rust control in North America over at least the last 50 years. I think we would say the evidence is that this has been oligogenic, or not simple monogenic resistance, certainly not complex polygenic resistance, just a handful of genes. And I'll give you a quote in a minute to illustrate that. Certainly in Australia, over the last 40 years, we've had sustained control of stem rust. And again, the genetic basis of that has been really what we would describe as being oligogenic. So here's a quote from Martins and Dick from Canada from 1989 about the long-term control of stem rust in North America. And basically you can see some of the names there of the genotypes they're mentioning appearing again that were mentioned in the previous presentation. Basically they said relatively few major genes in various combinations from 4 genetic sources provided control of stem rust in an epidemiological system comprising hundreds of millions of hectares and thousands of billions of rust propagules. Quite dramatic. And the other thing that I liked that I got out of that paper was that the often expressed view that so-called major genes are of fleeting value invites debate with respect to this system. Now, I think we would say that this example here does not just involve major genes. There are minor genes involved in this, very, very important ones such as SR2. But the point it illustrates is that there were relatively few genes that were deployed, and probably deployed very, very wisely, that have led to long-term widespread control of stem rust in North America. And the situation is very similar in Australia. So here's a list of varieties, wheat varieties that are currently grown in Australia, the most popular wheat varieties that are currently grown in Australia. Okay, so there are others, but these are the most popular ones. And I've given an indication there of the stem rust genes that occur in all of those varieties. And you can see some of them have 1, but there are some that even have 6. So we might say, well, if it's got 5 or 6 genes, this is a variety that has polygenic resistance, surely. But if we look at pathogen virulence, okay, so the ones that are now highlighted in red are the effective genes. The ones in black are the ones that our pathogen, a stem rust pathogen, have overcome. So now the number of effective genes in these varieties has come down to 1, 2, or 3. It's not really very many. You would consider this, as I said before, to be oligogenic resistance, certainly not the complex polygenic resistance that we often talk about where we're trying to stack 4 or 5 or even 6 genes. There's also another point there, and I think this is a really, really important point. There are combinations in most of these lines of major and minor genes. So the major genes there are in red. The minor genes, or genes we would consider to be minor, I've highlighted in blue. All of those varieties have a level of resistance, a very, very good or useful level of resistance to stem rust. And this is another important point. Just because they've got a resistance gene or more resistance genes doesn't necessarily mean they'll have a very high level of resistance. Some of these genes don't impart complete levels of resistance. But you can see there from those responses in the field, and these responses have come from a lot of work, that all of those varieties carry good levels of resistance to stem rust. So the points to note about this table really are, and this is one point I'd really like to make, I suppose, at the outset, is that in the last 40 years in Australia, we've only lost one stem rust gene, SR38. So I think that's a very, very good example of gene stewardship. We often hear, the people say, yes, we're putting genes out there, they're falling out, they're breaking down to the pathogen, we're losing these genes, we have to look after them. Well, here's a good example of looking after resistance genes. We've only lost one in 40 years. Resistances in those varieties, 1 to 3 genes based on various combinations of 7 effective known genes. There could well be a few others in there, but certainly the ones we know about, there are only 7, and the combinations are from between 1 and 3. Now, interestingly, of those 7 effective genes, we've actually had virulence in the past for 2 of them, SR30 and SR36, but we haven't picked up virulence for those 2 genes for a long time. So the virulence is no doubt out there, but it's at an extremely low level or extremely low frequency in the pathogen population. So in some ways, these genes have become very, very useful for us in combinations. It would be extremely silly to release varieties with only one of those 2 genes. There are some out there, you'll see Corel, for example, but in that case, farmers know that. They know it only has one of these genes. We're able to tell them that there has been virulence for this in the past and that they need to remain vigilant. The 2 most important things, and these are the 2 points I want to focus on in the rest of my presentation, is that if you look at the combination of genes in these varieties, they are mostly combinations of major and minor genes. And the other thing is that there are no rust suckers amongst them. And these are, as I said, are the 2 points I now want to focus on for the remainder of my presentation. So mostly combinations of major and minor genes. The evidence that we have, and it certainly is circumstantial, but there is now some experimental evidence that's emerging to support this idea, is that genetic background can actually influence the durability of resistance genes. So we can actually get a longer life out of genes if they're put in a background where there is a residual level of resistance imparted by, for example, minor genes. Good example of this that we have, and this is circumstantial evidence, but is the gene LR24 that was deployed in Australia. Now, it was used here for 17 years and it was used extensively, and yet it was effective. We finally detected virulence for LR24 in the year 2000. So we would have considered LR24 in Australia to be, by definition, a durable resistance. What we found was that LR24 was being released primarily in the north. What we found was when it was deployed in South Australia in 2 varieties, in Cricoff and Warrakatta, we detected the virulence very, very quickly. Both of these varieties were rendered VS, very susceptible by that mutation. That gene went and there was nothing behind it. Now, if you look at the situation in South Africa and in North America, LR24 was also deployed there, and it was spectacularly non-durable. So they had the virulence present even before the gene was used or very soon after. So there's 2 very contrasting examples of one gene. In Australia, we would have considered LR24 to be durable. We got a good 17 years out of that, and there were times when something like 40 to 50% of the acreage in the northeast of Australia was actually occupied or protected by that gene. But go to other countries, non-durable. So what's going on here? Well, we think the apparent durability, and this is sort of a retrospective view, but we think the apparent durability of LR24 in Australia is likely due to the presence of another gene, a minor gene, LR34, in the background. And those 2 varieties that were released in South Australia did not carry LR34, I'm sorry, so they had nothing in the background. So in this sense, we can think of this gene, this minor gene, which also gives one of these pleiotropic genes that also gives resistance to stem rust and to stripe rust and also to powdery mildew. We can almost think of this gene as being a gene steward. It's looking after the major genes that are sitting on top of it. And I think this is also what we've seen with stem rust in Australia. We've released varieties that carry important genes like SR2, minor genes that are sitting there behind the major genes, and we're getting an effect of those genes as gene stewards protecting the major genes. All right, so now coming to the rust suckers. This is the second point that I said I wanted to deal with in the last part of my presentation, and this is an area I think that really, that we push very hard in Australia and I think needs to get more attention when we're looking at large-scale disease control. An analogous example is vaccination in humans. And this is a quote that I read in a magazine back in 2010 about what the editor referred to as the herd immunity. And it comes back to this concept that Bob mentioned yesterday, this concern that Bob mentioned yesterday about complacency. We're seeing this with vaccination. People in Australia are opting not to have children vaccinated against whooping cough. This is an entirely preventable disease through vaccination, and yet people are not vaccinating children against it. And what's happening is that we're now seeing a resurgence in this disease again. And in 2009, 3 children in Australia died of whooping cough, an entirely preventable disease. So this is the herd immunity. If vaccination is going to work and it's going to stop these diseases, there's a threshold below which if people are not vaccinated, those diseases will make a comeback. And it's exactly the same, I think, this concept of herd immunity with rust control. If it's going to work, if we're going to use genetic control and durable resistance, and if we're going to have large-scale disease control, in a disease like rust that blows around over large distances on the wind, we need to have the majority of the farmers growing resistant varieties. We need that herd immunity. So this is just a very simple diagram, I suppose, emphasising that point. Very, very simplistically. It's like a compounding effect of rust control. We control rust, we end up with less rust. Less rust means lower pathogen diversity. The pathogen populations become stabilised in a sense. Our resistance genes then last longer, become more durable if you like, and then this feeds back into even better rust control. So this is the importance of people not growing highly rust-susceptible varieties. And it's a very strong message that we push in a communications program we have here in Australia called the Rust Bust. You'll see there's a booth outside the door with some of the publicity that we put out to growers, the extension material. We could be telling our farmers, only grow resistant varieties, make sure your varieties have 3, 4 resistance genes. The message is just too complex, and in reality, quite often, those options are not there for growers. They aren't varieties that fit that niche, that have that level of resistance. So we try to put a very, very simple message out. We say, don't grow very susceptible varieties. If you must, make sure you have a contingency plan in place. For example, fungicides ready to go, monitor your crop, and be ready just in case the rust develops. That's an example of one of the things we put out last year. It was estimated by one of the people in my group, I believe, that 1 hectare of wheat, 1 hectare of wheat can produce 19 kilograms of rust. So this is just, again, a simple message to illustrate to farmers how much growing a susceptible variety, the effect it can have. If you translate that, and I did some quick calculations the other day, to the situation we had in Ethiopia last year, where about 40,000 hectares of wheat was heavily infected with stem rust, and I know these are rubbery figures, but that would roughly equate to 760 tonnes of stem rust inoculum. And there are a number of ways of calculating mutation rates in rust, and I use 2 of them. And out of that amount of rust, we would have had between 28 and 8,000 million mutants in that rust population last year. Okay, so you can work that out in a genome size of 90 megabases. That's a lot of mutations. It's a mutation in every single gene, and that's not good. That's not good for gene stewardship. We talk about sexual recombination on barberry. Huge asexually reproducing populations also generate tremendous genetic variation. So growing these rust suckers, having lots of rust inoculum is a really bad thing for the industry as a whole. It's a bad thing for gene stewardship. Okay, so beyond, I've talked about continental examples of disease control. Beyond, let's say global. We can talk about durable resistance from the perspective of the host, but equally we can talk about it from the perspective of the pathogen. All right, so you can even define durable resistance as resistance to which pathogens find it very difficult to adapt. And we know that these rust pathogens move around. They blow around on the wind, they're moved around on people's clothing. We know in Australia, for example, that the frequency of the introduction of wheat rust isolates from other parts of the world is increasing as time goes by. We've seen in the durable rust resistance in wheat project, the Objective 23 surveillance work has done a wonderful job in mapping the spread of UG99 within the African continent and beyond. So if we're going to actually scale up and look at large-scale disease control beyond continents, I would say that one of the most important things we need to do is to have internationally coordinated monitoring of these rust pathogens. And that's what we've endeavoured to do in the durable rust resistance in wheat project, and I think with Dave Hodgson and the others involved in that work, it's been spectacularly successful in putting a system in place that can now be extended to other diseases so that we can have global monitoring to help with coordinated control efforts for these pathogens that move around the globe. Okay, so just to wind up, I think really when it comes to sustained disease control, and really this is what we're about. We need to be looking at controlling these pathogens, not just in our own backyard, but across the world, and we need to be doing this in a sustained manner. We've got examples of that. I showed those, 4 of those, but I think there's still a big gap in our knowledge as to how those examples have actually worked. There's a lot to learn. If we're looking at durable resistance and its role in that landscape or that broader scale disease control and also gene stewardship, I think we can't lose focus of the whole system. And again, Bob mentioned this yesterday when he talked about wheat breeding. Rust resistance is just one trait. And also the genetics is just one approach. So it has to be an integrated approach. We need to look at reducing overall rust inoculum levels to protect the genes that we're putting out there into agriculture. We need to look at what the genes do to epidemic development and epidemic onset. They all have different ways of expressing themselves, and that in turn has different effects on epidemic development. And that's important in the overall context of overall disease control. Gene stewardship, I think, very importantly means knowing the resistance genes present in our cultivars and breeding populations to ensure genetic diversity. And this also, in turn, allows us, hopefully, to avoid those rust suckers that are going to just build up the inoculum and threaten the genes that are out there. For sustained rust control at the continental level and beyond, this is where the rust pathogen monitoring, I think, really comes into its own and is really, really important. It depends on our willingness to share information, and we've been able to do that in the first and second phase of the durable rust program. I hope it continues in the future, but that's going to be very, very important, increasingly important, if these pathogens, if the frequency of their movement around the world increases. And then finally, just the concept, I think, of minor genes being used as stewards to look after the other genes that we're putting into varieties. So certainly, I think, whilst we all need to be responsible in the way we deploy these genes, and we all need to be good gene stewards, I think those minor genes can also help us in that regard. So, Callie, I'll leave it there. I guess we move straight on to the presentation now. If there's any comments, I'd be very happy to take them even during the break, if there's not time there. Thank you.